Dear students, welcome to E-Pathshala. Today we are going to discuss on the lesson F11 FN17, that is about minerals, selenium, zinc and calcium. Minerals and trace elements are very important for the human health, particularly the three elements which we are discussing today about the selenium, zinc and the calcium. For example, if you take selenium, this has been linked with many enzymes in the human body and has been shown a powerful antioxidant effects, and many good sources both in animal as well as plant sources. Particularly the selenium is also depends upon the soil content. Therefore, selenium rich soils naturally produces selenium rich foods. The another important element which we are going to discuss is about the zinc. Zinc is very well linked with other elements in the human body. Zinc is also linked with enzymes. Zinc is linked with immunocompetency of the at cellular level. So therefore zinc role has a multitude in human body. And the third aspect is about the calcium. Calcium role in the bone formation is very well described. Calcium levels in the human body approximately 9 to 9.5 milligrams per deciliter. Calcium rich sources are the dairy products, particularly you could take yogurt, milk, butter, cheese, and also from the plant sources you could find from the green leafy vegetables. When the calcium levels absorption is linked with the vitamin D in the circulation has been well established. Therefore, today we are going to discuss about these selenium, zinc and the calcium, their food sources, their biochemistry, the physiology, and at cellular level how these elements are interacting with other nutrients and performing the various functions in the human body which we are going to deal today. So we will talk about first selenium. Selenium is one of the important minerals in our daily diet, and it is present as small cluster of proteins called selenoproteins. The selenium content of soil in which the plant is grown is closely related to the selenium content in the plant foods grown in that soil. Plants convert selenium mainly into selenium methionine, that is SEMET, and incorporate it into the protein in the place of methionine. Selenium methionine can account for more than 50% of the total selenium content of the plant, whereas selenocysteine, methylselenecysteine and C-glutamyl-selenine methylcysteine are not significantly incorporated into plant protein and are at relatively low levels irrespective of the soil selenium content. In mammals, ingested selenium methionine is absorbed in the small intestine via the sodium-dependent neutral amino acid transport system. Now let us look at the bioavailability and selenium role in antioxidant protection. The group of enzymes called glutathione peroxidases require selenium for their regular activity. They play a major role in detoxification system and also to provide protection against oxidative stress. Of the 8 known glutathione peroxidase enzymes, 5 of them require selenium. In addition to the activity of glutathione peroxidase, selenium containing enzymes are involved in recycling of vitamin C from its spent from back to its active one, allowing for greater antioxidant protection. Approximately half of the characterized selenoproteins have been implicated to have antioxidant functions. Bioavailability is defined as the proportion of an ingested nutrient that is used for normal physiological functions or storage. So we look at this picture of this bioavailability and role in antioxidant protection of the selenium. Bioavailability is influenced by endogenous factors including growth, pregnancy or lactation, the efficiency of digestion, gut transient time, and the presence of gastrointestinal disorders or the disease. So like this, various factors will influence the bioavailability of any compound, and more so what we are discussing about the selenium. Although the proportion of the nutrient absorbed from the gastrointestinal tract is a major determinant for calcium and zinc, tissue utilization of the absorbed nutrient and renal concentrations of selenium are also important factors influencing bioavailability. Selenium bioavailability depends upon the conversion of absorbed selenium into a biologically active form and tissue retentions. However, because tRNA methionine does not discriminate between methionine and selenium methionine, a greater percentage of selenium methionine is incorporated non-specifically into body proteins in place of methionine in low methionine diet. As determined by GSH-PX activity in platelets, selenium methionine is absorbed and retained more efficiently than inorganic selenate or sodium selenite. Renal regulation is the mode by which whole body is controlled. Selenium in the form of selenium methionine is also significantly retained in proteins in the brain. What are the food sources of the selenium? The major sources of selenium in the world are plant foods, and the content depends upon the selenium present in the soil in which the plants are grown. Oysters, clams, liver, kidney, fish, and shellfish are rich sources of selenium. Animal meats also contain considerable amounts of selenium. Whole grains and seeds also contain good amounts of selenium. What are the functions of the selenium in our human body? Thyroid function. Selenium containing enzymes is responsible for transforming a less active thyroid hormone called T4 into the more active T3. The thyroid gland synthesizes two major hormones, triiodothyronine, that is T3, and the main biologically active thyroid hormone which occurs in liver, kidney, and thyroid, and thyroxine T4, which is produced solely by the thyroid and is biologically inactive. The control of thyroid hormone T3 is via DH, a selenoenzyme which produces most of the circulating T3 by deiodination of T4 in peripheral tissues. Deiodination of T4, that is triiodothyronine, is also catalyzed by type 2 5-deiodinase, which produces T3 primarily for local use and occurs in the central nervous system and the pituitary gland. However, irreversible damage of the thyroid gland caused by iodine supplementation to animals deficient in both iodine and selenium suggested that it resulted from an impaired selenium-dependent antioxidant system such as GSH-PX system. Impact of cooking, storage and processing on selenium. Selenium content of foods tend to be stable during storage. Animal foods tend to lose little selenium in cooking or processing. For example, the amount of selenium lost in the canning process of common seafood was marginal, less than 10% of the total pre-cooking amount. Processing whole grains is much more detrimental to selenium content. Relationship with other nutrients. A deficiency of both selenium and iodine can make thyroid disorders more severe than a deficiency of iodine alone. Deficiency. Selenium deficiency may contribute to the development of form of heart disease, hypothyroidism, and a weakened immune system. There is also evidence that selenium deficiency does not usually cause illness by itself. Rather, it can make the body more susceptible to illness caused by other nutritional, biochemical, or infectious stresses. Three specific diseases have been associated with selenium deficiency. Keshan disease, which results in an enlarged heart, and poor heart function occurs in selenium deficient children. Kashin-Beck disease, which results in osteoarthropathy. Myxomatous endemic cretinism, which results in mental retardation. Keshan disease was first described in the early 1930s in China and is still seen in large areas of Chinese countryside with the selenium poor soil. Dietary intake in these areas is less than 19 micrograms per day for men and less than 13 micrograms per day for women, significantly lower than the current RDA for selenium. Researchers believe that selenium deficient people infected with a specific virus are most likely to develop Keshan disease. Selenium deficiency has also been seen in people who rely on total parenteral nutrition as their sole source of nutrition. TPN is a method of feeding nutrients through an intravenous line to people whose digestive system do not function. It is important for total parenteral nutrition solutions to provide selenium in order to prevent a deficiency. Physicians can monitor the selenium status of individuals receiving total parenteral nutrition to make sure they are receiving adequate amounts. Severe gastrointestinal disorders may decrease the absorption of selenium, resulting in selenium depletion or deficiency. Gastrointestinal problems that impair selenium absorption usually affect absorption of other nutrients as well and require routine monitoring of nutritional status so that the appropriate medical and nutritional treatments can be provided. The next elements which we are going to discuss is about the calcium. Calcium is a key nutrient in the human body. The primary emphasis on calcium consumption during its initial scientific discovery was focused on early human life, primarily during growth periods of infancy and childhood. The interest on calcium requirements during the last decade has been expanded to apply to the entire life cycle from birth through elder years. Many commercial food and nutrition supplement products contain calcium fortification today in response to a wider audience. Calcium ions play a vital role in the physiology and biochemistry of organisms on the cell. They play an important role in signal transduction pathways where they act as a second messenger in neurotransmitter release from neurons in contraction of all muscle cell types and in fertilization. Many enzymes require calcium ions as a cofactor, those of the blood clotting cascade being notable examples. Extracellular calcium is also important for maintaining the potential difference across excitable cell membranes as well as proper bone formation. Calcium levels in mammals are tightly regulated, with bone acting as the major mineral storage site. Calcium ions are released from bone into the bloodstream under controlled conditions. Calcium is transported through the bloodstream as dissolved ions or bound to proteins such as serum albumin. Parathyroid hormone secreted by the parathyroid gland regulates the resorption of calcium from bone, reabsorption in the kidney back into the circulation, and increases in the activation of vitamin D3 to calcitriol. Calcitriol, the active form of vitamin D3, promotes absorption of calcium from the intestines and the mobilization of calcium ions from the bone matrix. Calcitonin secreted from the parafollicular cells of the thyroid gland also affects the calcium levels by opposing parathyroid hormone. Calcium storages are intracellular organelles that constantly accumulates calcium ions and release them during certain cellular events. Intracellular calcium storage include mitochondria and the endoplasmic reticulum. The role of calcium in human health. Calcium as a key nutrient is the most abundant stored nutrient in the human body. More than 99%, that is 1.2 to 1.4 kilograms, is stored in the bones and the teeth. Less than 1% is found in the extracellular serum calcium. When adults consume calcium as food or supplements, the average absorption rate is approximately 30%. The rate can vary widely due to multiple factors. For example, in pregnancy, When more calcium is required for the growing fetus, the calcium absorption rate increases. Calcium distribution in the body. Calcium is distributed among various tissues, compartments in the human body. The total serum pool of calcium, approximately 1200 to 1400 milligram, is very small. The extracellular pool maintains the plasma calcium level in a tight control at a constant serum level, typically 8.4 to 9.5 milligrams per deciliter, using a complex team of hormones and other substances. Serum calcium does not fluctuate with changes in dietary intake. The three organ systems are the kidney, the intestine, and the bone. The kidney is the primary mechanism for rapid release of or absorption of calcium through the filtration and urine excretion functions. Approximately 200 milligrams per day is typically excreted by adults through the kidneys via urine, but varies by diet and serum parameters. Functions of calcium. Calcium is used throughout the body in small amounts. Research has confirmed that calcium is involved in vascular contraction, vasodilation, muscle functions, nerve transmission, intracellular signaling, and hormonal secretion. Any change in serum calcium affects one or more of these functions. For example, hypocalcemia has been linked to higher risk of seizures due to its relationship with nerve transmission and intracellular signaling. Metabolic aspects of calcium absorption. Calcium absorption occurs throughout the gastrointestinal tract but varies by region. The majority of the calcium, approximately 65%, is absorbed where the pH is 6.5 to 7.5. That shows the importance of pH in the absorption of the calcium. In the ileum, the primary mechanism is passive absorption as the food moves slowly through this area of the gastrointestinal tract. It is important to note that calcium is not absorbed in the stomach. Calcium food sources. Predominant sources of calcium in the diet include dairy products like milk, yogurt, cheese, butter, etc. The most predominant food sources include dairy, but also anchovy, soy also. A wide variety of foods is necessary to achieve dietary guidelines. This reinforces the eating one single food or food group will not achieve dietary balance. Interaction of nutrients, calcium, phosphorus, vitamin D. It is important to remember that it is difficult, if not impossible, to discuss calcium alone. Calcium metabolism is a collaborative effort between calcium, phosphorus, and vitamin D and protein. Just like a musical orchestra, all of these nutrients are needed to create the end product, whether it is a beautiful song or a perfect bone matrix. So, in the combination of these nutrients, the products can be formed. Calcium metabolism interacts with phosphorus in healthy adults. Particularly during periods of growth, foods that contain both calcium and phosphorus can lead to positive effects on bone health. Effects of nutrient deficiencies of vitamin D and calcium. There are tissue-specific effects of inadequate calcium and vitamin D on both plasma serum, calcium, and vitamin D. The role of calcium sensing receptors that direct metabolic changes to maintain normal extracellular calcium levels is only beginning to be understood. Calcium sensing receptor cells are found primarily in the parathyroid glands but are also located in other areas of the body. They have been shown through research to detect even the smallest changes in the serum calcium and look for immediate sources of cellular calcium to restore plasma levels to the normal range. Low plasma vitamin D will also decrease enzymatic 25-hydroxy vitamin D3, 1-alpha hydroxylase activity. This in turn may change in normal differentiation and proliferation of bone and intestinal cells. Deficiencies in both dietary calcium and vitamin D will impair the function of these mechanisms and others to restore serum calcium. Long-term deficiencies set the stage for chronic diseases risk. Bone formation and maintenance changes throughout the life cycle. Bone is comprised of 60% mineral, 30% matrix and 10% water. A very simplified explanation of bone formation will be used to help understand the true complexity of this ongoing life cycle process. To create new bone or maintain bone composition, bone forming cells, that is osteoblasts, migrate into the non-mineralized matrix vesicles. This is followed by attraction of phosphorus and then calcium ions to create the mineralization of the bone. On the cellular level, sodium phosphorus transporter protein molecules creates the hydroxyapatite crystals as the precursors of the actual bone mineralization. Both phosphorus and calcium play essential interdependent roles in the formation of both new bone and repair of existing bone. The priorities of bone formation differ by population group. In children and young adults, emphasis is placed on strong baseline bone growth and structure as a foundation. By ages 25 to 30, the majority of the new bone formation is completed. After this growth period, emphasis is placed on maintaining the bone density and preventing bone loss. After the age of 50, research has shown bone density often decreases substantially, so the goal at this life cycle phase is to sustain bone mineral density and minimize bone loss. Progressive bone mineralization loss over time increases the risk of bone fracture and falling. The next important element which we are going to learn is about the zinc. Biochemical and physiological functions of the zinc. Zinc is ubiquitous within cells, in contrast to iron, which is contained in defined cellular components has defined physiological roles. The role of zinc in biology can be grouped into three general functional classes, namely catalytic, structural, and regulatory functions. Metabolism of the zinc. Zinc is absorbed in the small intestine by a carrier-mediated mechanism. Under normal physiologic conditions, transport process of uptake are saturated. The fraction of zinc absorbed is difficult to determine because zinc is also secreted into the gut. Zinc administered in aqueous solutions to fasting subjects is absorbed efficiently, 60 to 70%, whereas absorption from the solid diets is less efficient and varies dependent on zinc content and diet composition. Generally, 33% is accepted as the average zinc absorption in humans. Zinc is released from the food as free ions during digestion. These liberated ions may then bind to endogenously secreted ligands before they transport into the enterocytes into the duodenum and jejunum. Specific transport proteins may facilitate the passage of zinc across the cell membrane into the portal circulation. With the high intakes of zinc is also absorbed through a passive parallel paracellular route. The portal system carries absorbed zinc directly to the liver and then released into the systemic circulation for delivery to other tissues. About 70% of the zinc in circulation is bound to albumin, and any condition that alters serum albumin concentration can have a secondary effect on serum zinc levels too. Homeostasis. Maintaining a constant state of cellular zinc or homeostasis is essential for survival. In animals and humans, adjustment in total zinc absorption and endogenous intestinal excretion are the primary means of maintaining zinc homeostasis. The adjustment in gastrointestinal zinc absorption and endogenous excretions are synergistic. Shifts in the endogenous excretion appears to occur quickly with the changes in intake just above or below optimal intake, while the absorption of zinc responds more slowly, but it has the capacity to cope with large fluctuations in intake. With extremely low zinc intakes or with prolonged marginal intakes, Secondary homeostatic adjustments may augment the gastrointestinal changes. These secondary adjustments include changes in urinary zinc excretion, a shift in plasma zinc turnover rates, and possibly an avid retention of zinc released from selected tissues such as bone in other tissues to maintain function. Excretion. Loss of zinc through gastrointestinal tract accounts for approximately half of all zinc eliminated from the body. Considerable amount of zinc is secreted through the biliary and intestinal secretions, but most of it is reabsorbed. This is an important process in the regulation of zinc balance. Other routes of zinc excretion include urine and surface losses, desquamated skin, hair, sweat also. Thus, humans appear to have the capacity to regulate whole body zinc content over a tenfold change in intake as has been observed in experimental animals. Body zinc content is regulated by homeostatic mechanisms over a wide range of intake by changes in fractional absorption, normally 20 to 40%, and urinary 0.5 milligrams per day and intestinal 1 to 3 milligrams per day excretion. The bioavailability of the zinc refers to the fraction of intake that can be absorbed into the blood system and used for physiological functions of the body. For zinc, in healthy individuals, it is determined by three factors: the individual's zinc status, the total zinc content of the diet, and the availability of the soluble zinc from the diet's food components. If the individual's zinc status is discounted, zinc absorption is largely determined by its solubility in the intestinal lumen, which in turn is affected by the chemical form of zinc and the presence of specific inhibitors and enhancers of zinc absorption. Long-term zinc intake, that is zinc status, can also affect the absorption of dietary zinc. What are the inhibitors and enhancers of the zinc bioavailability? Various dietary factors can influence zinc absorption. Phytic acid, that is inositol hexane pentaphosphate, is the principal dietary factor known to limit zinc bioavailability by strongly binding zinc into the gastrointestinal tract. It is the major phosphorus storage compound in the plant seeds, especially cereals and legumes, and can account about 80% of the seed total phosphorus. Because of its high density of negatively charged phosphate groups, phytate forms mixed salts with mineral cations, which are assumed to play an important role in mineral storage. The potential interactions between iron and zinc has been a cause of concern. Proteins generally have positive influences on zinc absorption because zinc absorption tends to increase with the protein intake. Consumption of animal proteins, that is beef, eggs, and cheese sources, improve the bioavailability of zinc from plant food sources, possibly because amino acids released from the animal protein keep zinc in solution or in protein binds the phytate. Generally, binding of zinc to soluble ligands or chelators have a positive effect on zinc absorption as they increase the zinc solubility. What's the zinc deficiency? Organ systems known to be affected clinically by zinc deficiency states include the epidermal, gastrointestinal, central nervous, immune, skeletal, and reproductive systems. Clinical signs of severe zinc deficiency were identified in industrialized countries, notably in persons suffering from acrodermatitis enteropathica, a rare genetic disorder that specifically affects zinc absorption. Severe zinc deficiency resulting from other causes such as prolonged parenteral nutrition with inadequate zinc content produced similar clinical signs as in acrodermatitis enteropathica. Although less impressive in their clinical presentation, milder zinc deficiency is of numerically much greater importance. Moreover, most of the clinical features of acrodermatitis enteropathica were documented also in milder forms of zinc deficiency. Functional impairments identified in community-based trials may be more representative of mild or moderate deficiency. Some of these functional impairments are as follows. like in the growth and development. One of the most studied clinical features related to zinc deficiency is the impairment of physical growth and development. This effect is of most significance during the periods of rapid growth such as pregnancy, infancy, and puberty, during which zinc requirements are highest. The risk of infections, particularly the diarrhea, is characteristically, although not inevitably, a prominent feature of acrodermatitis enteropathica. Plausible explanations for a link between zinc deficiency and diarrhea include impairment of the immune system and intestinal mucosal cell transport. Relationship between zinc deficiency and age is also established. Furthermore, degenerative changes associated with aging may partly be due to zinc deficiency, including a decline in immunocompetency, delayed wound healing, and certain neurological and psychological changes. In general, clinical manifestations of zinc deficiency vary with age. In early infancy, diarrhea is a prominent symptom. Zinc deficiency also leads to impaired cognitive function, behavioral problems, impaired memory, learning disability, and neuronal atrophy. Skin problems become more frequent as the child grows older. Alopecia, growth retardation, and recurrent infections are common in school-aged children. Chronic non-healing skin ulcers and also recurrent infections are common among the elderly. What are the causes of zinc deficiency? The general causes of zinc deficiency include inadequate intake, increased requirements, malabsorption, increased losses, and impaired utilization. Inadequate dietary intake of absorbable zinc is the primary cause of zinc deficiency in most situations. This may result from low dietary intake or heavy reliance on foods with little or poorly absorbable zinc. Inadequate dietary zinc intake is common in many parts of the world. It is often exacerbated by physiologic conditions associated with elevated zinc requirements. Malabsorption of zinc may occur in a number of situations, for example, acrodermatitis enteropathica, malabsorption syndromes, and inflammatory diseases of the bowel resulting in poor absorption and loss of zinc may lead to secondary zinc deficiency, particularly in the presence of marginal dietary intake. Utilization of zinc is impaired in the presence of infected, as decreased circulation of zinc reduces the availability of zinc to the tissues. Fecal excretion of zinc is increased during the acute diarrhea. So, prevention of zinc deficiency. What are the intervention strategies? The major intervention strategies are dietary diversification or modification of the diet, supplementation, fortification, and biofortification. These strategies are not mutually exclusive but can be used in a complementary way. Dietary diversification or modification is a sustainable long-term approach to improving the intake of several nutrients simultaneously. Dietary diversification or modification strategies at the community or at household level have the potential to increase the intake of bioavailable zinc. Such strategies include agriculture intervention, production and promotion of animal source foods through animal husbandry or aquaculture, processing strategies at the commercial or household level to enhance zinc absorption from plant-based diets. Agriculture interventions focused on plant-based foods may have little impact on intake of bioavailable zinc. Milk and cheese are also important sources of dietary zinc. These foods generally have higher content of readily absorbable zinc than poultry, eggs, or fish. Nutrition education to promote dietary diversification or modification can lead to a greater intakes of animal source foods and thus bioavailable zinc. Supplementation is also another strategy. The supplementation programs are useful for targeting vulnerable population subgroups which are at a particular high risk of micronutrient deficiencies. The recommended zinc dosages are 5 mg per day for children between 7 months and 3 years and 10 mg per day for older children. When formulating multinutrient supplements, it is recommended that salts providing readily absorbable zinc like zinc sulfate, zinc gluconate, or zinc acetate are used because they are absorbed more efficiently. Fortification is a most cost-effective and sustainable strategy to overcome micronutrient malnutrition than supplementation. Fortification programs can also be specifically targeted to increase the intake of zinc in groups of high risks such as infants, and young children who consume particular type of food. In many countries, infant formulas and complementary foods are currently fortified with zinc and other micronutrients. Biofortification is also an important strategy. Plant breeding or genetic engineering strategies that either increases the level of zinc, reduce the content of inhibitors, that is phytate, or increasing the expression of compounds that enhance the zinc absorption, that is amino acids, have been considered to improve the bioavailability of zinc from plant foods. Biofortification differs from ordinary fortification because it focuses on intrinsic enrichment of micronutrients in plant pots that are used for food while the plants are still growing, rather than having nutrients from external resources and to the foods when they are being processed. This is an improvement on ordinary fortification when it comes to providing nutrients for the rural poor who are rarely have access to commercially fortified foods. As such, biofortification is seen an upcoming strategy for dealing with deficiencies of micronutrients in the developing world. Its additional benefits include higher yield where micronutrients are limiting plant growth and increasing vitality of seedlings emerging from zinc-enriched seeds. Thus, we have learned today of three important elements in the human metabolism. That is about selenium, calcium, and the zinc. So in conclusion, that we have observed that selenium is an important and essential element for the human body. Selenium comes from exogenous sources like from the plant foods as well as from the animal foods. Selenium has been shown for many health benefits, which includes the antioxidant activity. And the calcium which is an essential element, and with the dietary requirements also varied from age and physiological stage, gender, and adequate calcium is needed throughout the life cycle, which we have discussed. For example, if we take when the calcium levels are in lower, so the growth of the bone and also other cellular level cascade of events which are going to take place are going to be affected. And the zinc is also another important element which comes from the various sources, both plant as well as the animal sources, and their bioavailability is also inhibited, particularly when you have the inhibitors like phytates, which they can inhibit the zinc absorption also. So zinc is linked with immunocompetency, and of many enzymes are also zinc dependent enzymes are present in the system. Thus, what we have learned about these three elements are their sources, their physiology, their biochemistry, and the inhibitors and promoters of various factors which are present in the dietary system. That can be how we can remove that, or at least we can minimize that for improving the bioavailability by using the enhancers, and also various strategies how to overcome the deficiencies, like either by dietary diversification or by fortification or by biofortification or supplementation. We can overcome these deficiencies of these three important elements in the human body. Thank you.